I will talk a little bit about the interface between the hollow fiber model, its quantitative output, and the simulation and modeling that goes into it. So it's a large topic. So what we'll do is we'll use a few examples to go through it. I laid down what predictive modeling should be in my mind. So a disease model and a mathematical model should give a quantitative prediction. I don't think it's good enough to say this regimen is better than that regimen. We want to know how much is it better. What's the certainty? What's the uncertainty? We want to know those, how much response, from what dose? Because a regimen is made of drugs with certain doses. And how do we get to those doses? And accuracy should be judged based on clinical event rates and not another model. And this is, or even consensus. And here's why. It's because really what we're trying to model is what happens in patients. All models, you've heard this, all models are flawed, right? All models are flawed. What we really want to do is predict what happens in patients. And accuracy, therefore, should be based on clinical predictions. You've all heard about the hollow fiber model. This is the highly idealized version of it. It looks very different. And what it is a system of tubes. And this is the central aspect of it, which is called a hollow fiber cartridge. And essentially what you have here are thousands of semi-permeable membranes that allow us you can change the molecular cutoff point of these fibers. You can change the charge. You can change the surface area. What they allow us to do is if you take this cross-section, to have bacteria or macrophages or neutrophils with bacteria in this outside space called the peripheral compartment. And then what you do is you have media, liquid media. This is a liquid culture system flowing through there. And when you add your antibiotic to the system, you can vary the flow and dilution rates so that you can dial in any pharmacokinetics that you want. So peak concentration, clearance, trough concentrations. And what we have been able to do through computerized system for drug delivery is to also then produce different pharmacokinetics within the same hollow fiber system so that you can test combinations of regimens. So what do? You can then separate out, Veronique and some of the earlier speakers alluded to the different populations of Mycobacterium tuberculosis. So what you can do is you can put this whole system under hypoxia and then put Mycobacterium tuberculosis that's been under hypoxia, that's not growing, and do a study. Or you can have semi-dormant bacilli under acidic conditions. Or you can have these bacteria in log phase growth. So you can start mimicking the different populations of bacteria. And what it allows is quantitative output, not only bacterial numbers as you are giving drug, so how many bugs are alive, but how many are resistant. And of late, we've actually been able to overcome the high GC content problem in Mycobacterium tuberculosis and do RNA sequencing along the way as you give different combinations of drugs so that you have even more signal and more readout. So this is a slide that we use a lot just to show you that this was for moxifloxacin concentration time profile. The intended is this black line. And what you see when we sample at different time points is we can mimic fairly reasonably whatever concentration time profile we want. And as a matter of fact, once you have pharmacokinetics, say, in the lesion, right, this system allows you to dial, you can mimic any pharmacokinetic profile you want. Right. So even if it's in the lesion, we've been using epithelial lining fluid concentrations as a surrogate for some of the drugs for intramacrophage, for intra-lung concentrations. So I wanted actually to this is a list that is not exhaustive, but actually wanted to just put out a slide there to show that there were lots of things that we don't think of that were first seen in the hollow fiber that later on went to be confirmed in clinical trials and in other models. And if you go back, I throw this out and look at the dates at when these appeared. Some of these appeared well before the hollow fiber was popular, and then the clinical events and other studies confirmed this later. So there's a lot of them. So it's been very predictive. So I'll give a few examples of using the quantitative outcome and putting it into simulations. So you have your quantitative output on the relationship between changing concentration and microbial effect. And then you dial in human pharmacokinetics and even their variability, as you will see in one particular study I will show. And then you get so then what you do is you take this data and start doing modeling and simulations. Initially, mechanistic models on the relationship, differential equations on the relationship between drug concentrations and the effect with time, and later on to model, to project to populations of patients. And you do your simulations. And then you get different predictive outcomes. One is optimal dose, breakpoints, these are resistance breakpoints, microbial effect, resistance image, regimen performance, get an actual quantitative after you have gone through this. And what I don't show here is this goes back to the top, makes the model better. We go through that whole process so that the model, you continue learning as you are going through, and the predictive accuracy improves. So this is a slide a lot of you have seen in the past. This is a hollow fiber relationship between isoniazid effect and a microbial kill. And what these meaningless indices you see over here are just quantitative measures of drug exposure, and that what you have over there is the ratio of the AUC to the minimum inhibitory concentration. And and I can tell you at this stage, as you will see later, that in fact the hollow fiber and the mouse and the guinea pig, if you look at indices that are predictive of outcome, the patterns AUC to MIC have been fairly concordant on what's the pharmacodynamic parameter that's predictive of outcome. What's also fascinating about isoniazid is that if you look at the whole relationship between concentration and effect, it's fairly constant through clinical studies, through mouse studies, through guinea pig studies, very, very constant, so very deterministic relationship. So if you take this relationship and you say, now I want my optimal exposure to be what kills 80 or 90%, your EC80 or EC90, and you get that index, and then you say, you start playing around now and doing modeling. Now, how likely am I to achieve this if I gave a certain dose? Or, looked at the other way, what happens if I give a dose of 300 milligrams, the standard dose? How likely, or where are patients likely to fall on this concentration effect curve? So you do simulations for that. So this was one of the earlier ones we did, and this was part of our benchmarking. So what we did is, isoniazid is fun for modelers because 88% of variability is driven by certain SNPs of certain enzymes. Acetylation phenotype as we measure it, but now we have a very good idea of the genotype. And so what you do is, so you can predict your pharmacokinetics much better for isoniazid than a lot of drugs. And now these acetylation phenotypes, the percentage of patients varies depending on where you are, like all things. In Cape Town, Hong Kong, and Chennai, and we chose these because there were clinical studies that had been done in these places, and we also had a good idea of the MIC distributions. Then we took population pharmacokinetic data of isoniazid and put in all this variation, this variability, and then said, if you got a dose of 300 milligrams of isoniazid, and you were in any one of those areas, what was your microbial kill going to be like? And so this is given as EBA, and this is actually what was observed in clinical trials in different places. You can actually see that it's not a very bad predictive model. You can predict what happens in patients, probably to 2 decimal places, the rates of kill. with a dose. I'm actually going to zoom through. I just got told that the time is expiring, and I wanted to talk about what was predicted before and seen after, as the word predict means. And so here this was a combination therapy study, and we had been looking at factors associated with failure of therapy and resistance emergence. And it became obvious to us that pharmacokinetics were probably more important than adherence in particular failure associated in this population in the Western Cape that we simulated based on hollow fiber results. And what we did here, this is a simulation of what happens in sputum based on hollow fiber findings and other things. What happens in sputum given the pharmacokinetic variability of the three drugs that are given, the three standard rifampin, isoniazid, and pyrazinamide. And this is what you would predict the sputum falls for 10,000 patients in Western Cape would be, with a sputum conversion rate of 56%. As you can see, there is a wide uncertainty. And in fact, I can tell you that clinical studies from the Western Cape show the 2-month sputum conversion rate right around that. What was even more exciting was we were able to find out that a lot of patients would have very low drug concentration, 1 or 2 or 3 of them. And that these, if you followed them, led to drug resistance. You would have drug resistance predicted to arise in 2 months of about 0.7% of all patients. So what we did is we published this data and set it out, and then went ahead and did a clinical study. And I'm going to finish now so this Dr. Helen McIleron and the guys in Cape Town, prospective study of 142 patients. PKs were done on these. Patients were followed for 2 years. Long story short, 2 months in hospital, then PK is done and followed. So if you did nonlinear interactions, and classification and regression tree analysis is what we used, and you said, so what were the top 3 predictors of long-term outcome? If you put in lung cavities, non-adherence, all clinical factors, and PKs, the top 3 were a pyrazinamide AUC above this threshold, followed by rifampin, followed by isoniazid, the AUCs. And it turns out, in fact, the hollow fiber tells you that the AUCs of these drugs are going to be important. And for those patients in whom we had concentration, in whom we had MICs, you got the exact AUC to MIC indices you got in the hollow fiber predicting outcome. So I left out one slide, which is at 2 months. The rate of resistance emergence in the patients in Cape Town, given this pharmacokinetics, was 0.7%. Virtually identical. I'll stop here and take questions.